In this video, we're going to talk about the stages of meiosis, which is the production of gametes or sex cells. To recap types of reproduction, there's two forms. There's asexual reproduction, so only one parent is required, and mitosis is used. So exact copies are made, and an example of something that would reproduce by asexual reproduction will be bacterial cells. Sexual reproduction is when there are two parents and there are genetically distinct offspring made by those two parents, except in the case of identical twins or triplets or so on. So the process of meiosis occurs in all sexually reproducing organisms, and it only occurs in one part of their body, in the reproductive organs, so either the ovaries or the testes. And the end result is to produce sperm or egg cells. So those are the sex cells or the gametes. And each human sex cell contains half the number of chromosomes found in a somatic or body cell. So instead of having 46 chromosomes, they have 23 chromosomes. So you might question, why do we have meiosis in the first place? Well, we have to reduce the chromosome number to be able to retain the right number of chromosomes in the next generation of the offspring. So when an egg and a sperm unite, the correct number of chromosomes is regained, 46, and then we've restored the right number of chromosomes for the next generation. And we've introduced more genetic diversity. So what are haploid cells? Well, haploid cells have a single set of chromosomes, and we represent them with the symbol small n. So eggs and sperm they're haploid, and they have the symbol n. So 23 single chromosomes in a human egg or sperm cell. Diploid cells, on the other hand, have a double set of chromosomes. So we represent that with 2n. So the example would be, we have 46 single chromosomes in a body cell or somatic cell, or we say we have 23 pairs. So there are three main steps of meiosis. The first is replication. So we produce an exact copy of all the chromatin or all the genetic material. So for a temporary moment in time, we go from 2n to actually 4n. So we've doubled each of those chromosomes. And basically what happens, we always represent chromosomes looking like an X, like that. There's actually two chromosomes. This is the doubled chromosome after the DNA has doubled. So that would be when things are 4n. And then when they pull apart, they're 2n again. The next step would be meiosis. So the chromatin copies are divided 2 times instead of 1 time. That's the difference between mitosis and meiosis. And they're randomly distributed to the 4 completely different haploid nuclei. And then the final step is cytokinesis, and that's when the cell cytoplasm divides into 4 parts. And in sperm cells versus egg cells, the division of that cytoplasm is not even. We'll talk about that a little later. So just like in mitosis, there is an interphase stage in meiosis, and that's just the growth and synthesis phase. And that's when the chromosomes replicate, and they result in pairs of identical sister chromatids in the beginning of meiosis. This means at the beginning of meiosis, there are 46 doubled chromosomes or 92 single ones. So again, this is a doubled chromosome. This is a single chromosome. So these two halves of the X are the sister chromatids. The whole thing is the chromosome, and it's been doubled. This is also a chromosome. It just hasn't been doubled. It hasn't replicated. Another term to bring in is tetrad. So a tetrad is 2 homologous pairs of sister chromatids. So it gets a little complicated. So again, these 2 orange parts are the sister chromatids. The orange and the blue are the homologous chromosomes. And then the whole thing together is the tetrad. So again, imagine this is chromosome number 1. So you've got 1 from your dad, 1 from your mom, and then they replicated. So now each of those has two halves that are identical. So these two sides are identical. These two sides are identical. But these two chromosomes are similar but not the same because one came from your dad and one came from your mom. But all together, the whole thing is a tetrad. So again, as I mentioned before, meiosis has to happen twice. There has to be two divisions. So we're going to go through the stages of meiosis 1 and then quickly just recap meiosis 2. So we have prophase 1. And in prophase 1, the chromatin condenses into visible chromosomes. So you can see they're condensing. So here they are. The homologous pairs find each other and they form tetrads. So there's one homologous pair. There's another homologous pair. That process is called synapsis. We have 23 pairs of homologous chromosomes, as I mentioned several times already. One pair came from your mom and one pair came from your dad. And during this stage, centrioles begin to move to the opposite ends of the cell. We can't see them here, but they would be moving to those parts. And the nuclear membrane is dissolving. You can see that breaking down in this transition. During prophase 1, we also have a process called crossing over. Because the tetrads pair up and they overlap and they line up perfectly, the DNA from each of those will overlap in certain parts and they'll actually swap their DNA. So this will actually cause more genetic variation. So this red one, we'll imagine, originally came from your mom, and the blue one originally came from your dad, and these two sister chromatids overlapped one another. So the piece of DNA from your dad went to your mom's chromosome, and that piece from your mom's chromosome went to your dad's. So then you see this, like so. Then we move on to metaphase 1. So those tetrads, or pairs of homologous chromosomes, they line up along the equator. So M for middle. P I always think of as prepare, prophase for prepare. M for middle, so the homologous chromosomes, the tetrads, line up along the equator in the middle of the cell. And how they orient is random. So half of them might be from your mom on the left and half might be from your dad on the right and so on. So there's a 50% chance for the daughter cell, each daughter cell, to get either the mom's or the father's homologue for each chromosome. So that's all random in how they line up. They just line up however they choose. Then, from the centrioles to those centromeres, spindle fibers form. So let's just redraw a chromosome here. So this is the centromere. You would have learned that last year too. And then at either end of the cell are those centrioles. And they're going to put spindle fibers attaching each, almost like a fishing line. And eventually they'll pull the pairs of chromosomes apart. The next step is anaphase 1. So the centromeres at this point aren't breaking apart. Instead, the homologous chromosomes are separating and moving to opposite poles of the cell. So we're still taking doubled chromosomes. They haven't split apart. So half the doubled chromosomes are going to one side and half the doubled chromosomes are going to the other side. So we end up with 23 doubled chromosomes in each cell. And again, we'll stress the random assortment of chromosomes. So during anaphase 1, the double-stranded chromosomes separate and move to the opposite ends of the cell. And those chromosomes will be sorted randomly. So you can see that happening here in each of these. So this is meiosis 1, and this is meiosis 2. We're going to talk about that later. So then in telophase 1, the spindle fibers disappear. Cytokinesis takes place and you end up with 2 cells with 23 doubled chromosomes. We still need to split those doubled chromosomes through this next step of meiosis. So meiosis 2, the stages are exactly the same as mitosis. So both cells from meiosis 1 will go through the stages of meiosis 2. So it'll have prophase 2 where they'll prepare, the nuclear membrane will start to dissolve. Then metaphase 2, the chromosomes will line up along the equator. And then in anaphase 2, the centromeres divide and the single chromosomes will go to opposite ends of the cell. So at the end of meiosis 2, the result is 4 cells with 23 single chromosomes in each. Now in class, we're going to do an activity where you'll actually model that and you'll actually go through step by step the stages of meiosis 2. So stay tuned for that.